Today we are going to discuss about two things that is related to the transmission of impulse. It is excitatory postsynaptic potential and inhibitory postsynaptic potential. First, I will define what is excitatory and what is inhibitory postsynaptic potential. That is inhibitory or excitatory excitatory means excitatory postsynaptic potential is such a potential which makes the neuronal membrane more likely to generate action potential. So it causes the conduction or the opening of more and more sodium gates. So more and more sodium gates will open up, and this causes the action potential to generate very easily. So excitatory postsynaptic potential is such a potential generated by certain neurotransmitters which make the postsynaptic membrane more likely to produce action potential. We will discuss all these things after this. And the second point is that such a potential or such a neurotransmitters, they hyperpolarize the membrane. And what will they do? They hyperpolarize they not hyperpolarize, they depolarize. They depolarize the membrane easily. And the second point, third, is that they will bring resting membrane potential nearer to threshold. They will bring the resting membrane potential nearer to the threshold. And such few examples of the excitatory postsynaptic neurotransmitters. And the third point is that they open up more sodium gates, sodium gate channels. Such neurotransmitters which produce excitatory postsynaptic potentials, they are acetylcholine, glutamate and aspartate. Examples are acetylcholine, aspartate, aspartate and glutamate. And many other neurotransmitters. These neurotransmitters are such type of the neurotransmitters which generate such an excitatory postsynaptic potential which causes the more and more opening of the sodium gates. When more and more sodium gates will open up, the membrane will easily depolarize and generate action potential. So excitatory postsynaptic potential is such a potential which generated by certain neurotransmitters which makes the postsynaptic membrane more likely to produce action potential. So this is about the excitatory postsynaptic. Now we will discuss about the inhibitory postsynaptic. Inhibitory postsynaptic potential is such a potential postsynaptic membrane which are produced by certain other type of neurotransmitters. I'm not writing here the neurotransmitters, I will just define. So EPSP is such a potential at postsynaptic membrane which carry away the resting membrane potential from threshold. And they will make the postsynaptic membrane less likely to generate action potential. So what will they do? How they inhibit the resting membrane potential to generate action potential? They will open up chloride ion gates and inhibit opening of more or fast sodium ion gated channels, or simply sodium channels. So for examples of inhibitory postsynaptic potentials are GABA and glycine, which are the two main important. It is glycine and GABA. These are the neurotransmitters which make the postsynaptic membrane less likely to generate action potential. So let us go through the proper procedure that how they generate action potential and how they inhibit to generate an action potential. Before defining or before go through the process of action potential generation, I will define a certain thing. That is, for example, this is the presynaptic membrane which is responsible for the carrying of impulse from the neuron and convert at the neuronal ending into neurotransmitters or mechanical message, and this mechanical message is then released by the vesicles to the postsynaptic membrane. First, these neurotransmitters releases into the postsynaptic or synaptic cleft, and after here, when this neurotransmitter reached to the membrane of the postsynaptic neuron, there are the certain receptors are here which receive the neurotransmitters and generate action potential. So before going through this, we will discuss that whenever more and more sodium ions open up, more sodium will enter inside the cell membrane of this neuron and generate action potential. So the conductance is directly dependent upon the number of sodium gates opening here. So more sodium channels open up, more action potential is generated, and the conductance is very efficient. So more conductance is produced. If this number of gates is less, there is sodium channels are less, so less sodium will come inside and it will generate action potential which will not be very efficient, and those type of neurotransmitters which stop the opening of certain type of or more efficient sodium ions and causes the opening of some other channels for negative ions, for example chlorine. So they will make the membrane hyperpolarized and less likely generate the action potential. So same procedure will carry or here. For example, this I will consider this one acetylcholine and this one GABA. So they will perform separate actions. So whenever this acetylcholine releases from the presynaptic membrane and come down to the postsynaptic membrane, their receptors are here and they will sense the presence of acetylcholine and they will open up sodium channels. So when more and more sodium channels open up, this is the acetylcholine binding site and these are the gates, sodium gates. So what they will do, they will open up more sodium channels more efficiently. As more and more sodium channels open up, more sodium rushes inside. More and more sodium will move inside. And this entry of sodium ions into the membrane make the membrane depolarized, that is positive inside and negative outside. So they will do repolarization. If we make the graph, then we will make such a graph. For example, this is the graph and this is the resting membrane potential. Suppose this is the resting membrane potential or minus 70 to minus 90. This is the resting membrane potential. Whenever more and more sodium will come inside, first the membrane which was at the rest, they will move towards the threshold. If the efflux of the movement of the sodium ion is so efficient that it reach to the threshold, it will produce action potential. This will be the graph made by the acetylcholine, that whenever the acetylcholine attached to the sodium gates, the sodium gates will open, more and more sodium gate will open up, and first the mechanically stimulated sodium gates open, and after this the voltage gate sodium open, and this entry of the opening of these gates will stimulate the sodiums to rush inside, and more sodium will come inside and make the membrane depolarize, and here what will cause, it will generate action potential. That is generate action potential. So what will they do? That the action potential is generated. So we can say that the postsynaptic membrane become excited. So this potential is known as excitatory potential. So the acetylcholine is generating excitatory postsynaptic potential, and this excitatory postsynaptic potential make the postsynaptic membrane depolarize and less and more likely to generate the action potential. So it will generate the action potential very efficiently. If we look in case of this GABA, then what will happen whenever GABA reach and this GABA is accepted by the receptors present on the postsynaptic membrane, they will open up other type of gates and less efficiently open sodium gates. So less sodium gate will open up and more other gates will open up, and these gates what we called as the gates for negative ions, and this is for the sodium. So I will make a different color here. So less sodium ion will open up sodium ion gate, and more negative gates open up. So more and more chloride ion get inside, and less sodium will in inside. Is if we make a graph of this, then what will we make? We make such a graph. This is minus 70 millivolt, the resting membrane potential, and threshold potential. So whenever acetylcholine reach here and receive the membrane depolarize to the threshold and generate an action potential. Whenever GABA reached here, these neurotransmitters, they will accept by the gates which are sensitive or which are for the chloride ions. They will open up in more and more chloride inside and less sodium inside. Due to the entry of this negative ion inside, the membrane which was already at minus 70, they will starts to hyperpolarize. So they will not generate action potential because more and more chlorine is coming inside. So more and more chlorine come inside and make the membrane more hyperpolarized, and this hyperpolarization carry this resting membrane potential away from the threshold. The acetylcholine which making or carrying the with the membrane potential, that is resting membrane potential, toward the threshold, and here it is carrying the resting membrane potential away from the threshold and make it hyperpolarized. So it will hyperpolarize the resting membrane potential and inhibit the postsynaptic membrane to generate action potential. So it will stop the passage of impulse, and it will accelerate the passage of impulses. So here 2 points clear that neurotransmitters are different type of neurotransmitters. Certain type of neurotransmitters which causes the postsynaptic membrane to generate action potential, well, and they are called excited postsynaptic potentials, and such type of potential which is created by the certain type of neurotransmitters known as GABA, glycine, they will open up more chloride ion channels, and the entry of the more negative ion inside make the resting membrane potential more negative. So this causes the hyperpolarization and carry the resting membrane potential away from the threshold, and it will causes the membrane less likely to generate the action potential. And GABA, which inhibit the transmission of impulses, and acetylcholine, which promote the transmission of impulses. So this is about all about the excitatory and inhibitory postsynaptic potential. It is not so difficult, and if you remember these points, that acetylcholine and GABA open up the more and more sodium ions in GABA will open up more chloride ion, and it will be very easy to remember. Thank you.